final presentation of the webinar series. Today I'm going to switch off my camera just to save some bandwidth. So as Colette mentioned, today we'll be talking about the NovaSeq 6000. So for you that don't know, this is probably the most exciting sequencer that you'll ever meet in your entire life. This is a very, very big instrument, and I'm really excited to tell you more about it. So just to give you a bit of an overview of all of the systems that we haven't discussed all the systems, but some of the systems we have discussed and some of the applications on the systems. So if we look at our lowest throughput targeted sequencing methods, we would go for something like a mini-seq, MiSeq, or a NextSeq 500 or 550. Our higher throughput methods, we can use instruments like the NextSeq 550 as well as the NextSeq 1000 or 2000. And then for your personal scale, so really low throughput applications, we can use the iSeq 100. And then comes in the beast for production scale, which is the NovaSeq 6000. So the NovaSeq 6000 has proven architecture. It's widely published. It's an industry-leading platform built for scalability with better data economics, removing the barriers to answering your biggest biological questions. It's got flexible performance because it's a really highly figurable system that supports the broadest range of methods at any scale. It's got immense discovery power, and you can sequence even deeper into genomes. And you've also got the streamlined operation where the system is designed to increase your lab efficiency with a simplified workflow and seamless user experience. So some of the features of the NovaSeq 6000 include the high density patterned flow cell. So we do, with the high density, the patterned flow cell, we have optimized data management because you have the 8 times an increase in the primary analysis speed, and it also decreases your data footprint by 25%. With the NovaSeq flow cells, we've also got new surface chemistry. So you increase the signal to noise ratio by having smaller clusters with these high density patterned flow cells, and having smaller clusters, you can have also brighter clusters. They've also engineered the nucleotide dyes compared to our previous systems. We've also got new and superior imaging technology with up to a 4 times faster scanning rate. And what is really nice about the NovaSeq is the fact that we've got the dual run mode. So you can actually put 2 flow cells simultaneously on the NovaSeq 6000. You can run either 1 or 2, and you can mix and match different flow cell types. So therefore you can run different studies in parallel. And then also a very nice feature is the fact that you can actually split the lanes, which is not possible in any of our other systems. So the flow cells, all the flow cells has 2 lanes, except the S4 flow cell has 4 lanes. And we can use the XP workflow, which is basically a dock where you physically split the lanes, which can also increase your efficiency and flexibility on the system. So Peter did talk about the pricing structure per gigabase and what it would cost you per gigabase on the different systems. So obviously, the cheaper the system and the lower the throughput of the system, the more expensive it is to sequence. And the larger the system, the cheaper it becomes. So if you look at something like a MiSeq, which only has 15 gigabases of data, you look at about 2,500 Rand per gigabase. If you go then up to the NextSeq 550, it costs about 850 per gigabase. The NextSeq 2000 has a 360 gigabase output, which costs about 360 per gigabase. And then on the NovaSeq 6000, we have 6 terabases of data. So that's 300 terabases per flow cell on the S4 flow cells. And that would cost you approximately 100 Rand per gigabase. So with something like the NovaSeq, we can run more samples. There's a variety of new applications that you can run on the NovaSeq, and you can increase the depth of sequencing. So if we look at the configuration of the NovaSeq, we've got 4 different types of flow cells. So the smallest flow cell is your S' or the SP flow cell, followed by your S1, S2, and then the largest one is your S4 flow cell. So what differs between these flow cells is obviously the output of the flow cell. So your SP flow cell has the smallest output ranging from 65 to 400 gigabase, and the output obviously depends on the read length of your kit. And then on the S4 flow cell, per flow cell, we have up to 3,000 gigabases of data that can be generated. Yeah, and then the runtime obviously also differs between your smaller and your larger flow cells, but you can run an S4 flow cell with 300 gigabases of data in approximately 44 hours. So if we look at the estimated sample throughput for some of the key applications, we've got human whole genome sequencing. If we do that, if we generate about 120 gigabases data per sample with a 30 times genome coverage, we can sequence 2 samples on the SP flow cell, 4 on the S1, 10 on the S2, and up to 24 samples on the S4 flow cell. Meaning if we do this in dual mode, we can sequence 48 samples, whole genome sequences, human whole genome sequences per run in approximately 44 hours. For whole exome sequencing, the general guideline is about 100x coverage for the whole exomes, and there we generate 8 gigabases data per sample, and there you can range anything between 20 to 250 samples between the different flow cells. And then if we look at transcriptome sequencing, for higher discovery, we generally suggest more than or about 50 million reads per sample, and there it will vary anything between 16 samples to up to 200 samples on the S4 flow cell. And this is, as I said, this is per flow cell, so you can double these numbers if you were running 2 flow cells in one go. So if we look at the NovaSeq 6000's applications, it really has the widest variety of applications available, and basically that's due to the high throughput of the system. So here we can sequence multiple human whole genomes. We can do deep metagenomic sequencing. We can sequence exomes. We can do circulating tumor DNA sequencing, chromatin accessibility, methylation profiling, spatial transcriptomics, RNA sequencing, and enrichment studies. And today I would like to focus on these 3 applications. So we're going to look at human whole genome sequencing, then RNA sequencing, so more transcriptomics, and then your circulating tumor DNA sequencing for cancer applications. So looking at whole genome sequencing, Peter discussed exome sequencing on the NextSeq 2000 last week, and you might wonder, why would I want to do genome sequencing rather than exome sequencing? So both has got their places. As you might know, the whole exome sequencing, they target the protein coding regions of your genome. So it is a reliable and sensitive way to detect coding variants, and it's a really fast and cost-effective way of sequencing. So on average, an exome size is about 45 million bases, and if you do that 100 times coverage, you generate about 8 gigabases of data. Now, if we look at the whole genome of a human, we're looking at approximately 3.2 billion bases. We can cover the whole genome with about 99.9 sensitivity with 30 times coverage, and we generate 120 gigabases of data per sample. So this is a really comprehensive view of the genome, and we look at both the coding and the non-coding as well as mitochondrial DNA. It's also very reliable and sensitive detection of all variant types, and it's relatively low cost. And these days we've got really fast library preparation kits for this application. So looking at the workflows when we compare genome versus exome sequencing, we'll start off with genomic DNA, which we then have to put through a DNA fragmentation. So when we fragment, we have a pool of introns and exons, and this is where the two methods will split. So with whole exome sequencing, we will hybridize and we will design probes, and they will hybridize and capture your exons. And then we do a bead pull down with streptavidin beads, and we wash away everything that hasn't bound to the beads. So these captured exons, we then PCR amplify to make more of them, and then we can start sequencing them. With whole genome sequencing, we have both PCR and PCR-free library preps. If we look at the PCR-free library preps, we don't have to amplify anything. We look at both the introns and the exons. We can sequence that directly. And then we go through the analysis and interpretation of that. So we do the mapping, we do the alignment and variant calling. We do the variant annotation, and then we will filter these variants and interpret it with a final report in the end. This is just a bit of a comparison between different methods available for genetic testing. So the old way of doing the testing was via standard sequencing. Then we did some targeted NGS. PCR is also a widely used method. But if you look at whole genome sequencing, we can cover all of these things. So unlike other methods, the whole genome sequence methods are really robust, and they capture all the common variant types. It also captures the copy number variants with greater resolution than something like chromosomal microarrays, and it also captures variants in exomes with much greater accuracy than whole exome sequencing. Whole genome sequencing is technologically much simpler than whole exome sequencing, and it's also got a much faster turnaround time. So with the workflow for large whole genome sequencing, we obviously have to do a library prep first, then we sequence, and then we analyze the data. So for the library preparation, we've got four different library preparation kits. So we've got the Illumina DNA PCR-free prep fragmentation kit. So this is one of the newest kits that's been released. And then we've got the Illumina DNA prep fragmentation. So this is with PCR. And then our older legacy kits were called TruSeq DNA PCR-free and TruSeq Nano. Then we have to choose a sequencer. For whole genome sequencing, you can technically do one whole genome sequence at 30 times coverage on a NextSeq 550. That would not be very cost effective. So therefore, we would recommend that you do it on something like a NovaSeq 6000 system. And after that, you would go through the analysis pipelines, which there are many pipelines available on base-based sequencing hub, such as the Dragon germline application. So if we look at DNA fragmentation approaches after we've extracted our DNA, there's three different approaches that we can follow. So we can either do mechanical fragmentation, we can do chemical or heat fragmentation, and lastly, we can do enzymatic fragmentation. So mechanical fragmentation, it's very robust and it's got low bias. However, you do need a Cuvaris for this application, and a Cuvaris is pretty expensive piece of equipment, and it's very challenging to try and automate the mechanical fragmentation. So chemical and heat fragmentation on the other side, you don't need any specialized equipment. It's really easy to automate. However, this is only applicable for RNA, and it may require some optimization. Whereas with enzymatic fragmentation, you also don't need any specialized equipment. It's really easy to automate. However, it can be a bit sensitive to your DNA concentration inputs, and it may require some optimization. So to mitigate these last 2 points on enzymatic fragmentation, Illumina introduced the Fragmentation library prep kits. So the Fragmentation library prep kits work with bead-linked transposons, and I'll explain that in the next slide a little bit more. But you would have these magnetic beads with your transposons, add your DNA, they will add tags, and you will go through the indexing process. Please notice that there's no PCR involved in this step. So comparing the new Illumina DNA-free PCR kit, the Illumina DNA PCR-free kit to the TruSeq DNA PCR-free kit, which is the older type of kit, we've got a wider range of input material. So with the newer kit, we can use your genomic DNA, we can use blood directly, we can use saliva directly, we can use plasmids, and we can also use dried blood spots. So there are specific additional protocols to use these extra input types. With your input amount, you can go as low as 25 nanogram up to 1 microgram of gDNA. And with the older kits, you needed quite a lot of DNA to sequence, 1 to 2 micrograms. The fragmentation method, as I explained, is on bead with the bead-linked transposons. And with the TruSeq kits, you need that Guevara's sonication. We've also got 384 dual indexes, so you can multiplex 384 different samples. With the older kits, you could only multiplex 96 samples because we only had 96 dual indexes available. These kits are supported on most of our systems at Illumina, and a big plus point of this kit is the fact that the total work time has been reduced dramatically. So you've got basically 90 minutes on hands-on time with your extracted genomic DNA and about a 3-hour workaround time with your raw blood and saliva. So just to explain the bead-linked transposon workflow a little bit better for you that don't know. So all it is, it's a magnetic bead, and it's got these transposons linked to the magnetic bead. When you add your DNA, it does fragmentation. So fragmentation is, it fragments the DNA and it also tags it. And the tag is the tag for the adapters to bind to. And in the end, you have these DNA fragments that bind and saturate the magnetic bead. So the fragmentation of the DNA is not time dependent, creating a way more robust workflow. And immobilization of the transposon yields then your consistent insert sizes across various input concentrations. And therefore the saturation allows for integrated extraction and a quant-free final library prep. So it's already normalized. You've got the consistent library yields, consistent insert sizes, and you can imagine if you have so many samples that you have to normalize at the end of the library prep, it becomes quite tedious, which is not necessary with these bead-linked transposons. So when we look at the PCR-free library prep kit from Illumina, these kits, if you compare it to the Illumina, the normal DNA prep kit with PCR, the PCR-free kit reduces polymerase slippage and therefore increases your indel calling accuracy. And you'll also see that therefore the PCR-free kit decreases the gaps in your final data and therefore reduces any false single nucleotide variant calls in the genome. So if we look at specifically on the NovaSeq 6000 for whole genome sequencing, again with 120 gigabases of data per sample at 30 times coverage, we can do anything from 2 samples on the SP flow cell up to 24 samples on the S4 flow cell. And an interesting fact is if you look at the cost per genome, what it cost in 2001, up to $100 million. And this graph is actually quite old because we've recently launched the new V1 kit on the NovaSeq, where you can sequence a whole genome at 30 times coverage for about $600. So the second application I would like to discuss is transcriptomics. So why would we want to do bulk RNA sequencing? So some of the key applications for RNA sequencing is to look at genetic or gene expression profiling, to discover new transcriptomes, and to also detect other transcripts. The advantages of RNA-seq compared to something like microarray technology is the fact that it's hypothesis-free. You don't need any oligos pre-designed and bound on a microarray. The discovery power of transcriptome or RNA sequencing is immense. You've got very high throughput capabilities, especially on the NovaSeq. You get the sequence and variant information, and there's a very high specificity and accuracy. And RNA sequencing is applicable in numerous fields, including oncology, genetic disease research, infectious disease research, cellular and molecular biology, microbiology, as well as epigenomics. So you might ask different questions, research questions, and these questions you would choose a specific kit for your application. So you might ask which coding genes are expressed in the sample. So therefore, we would look only at the coding transcriptome assembly. Therefore, we would do something like mRNA sequencing, the messenger RNA. If we want to know all RNA transcripts are expressed in a sample, then we would do total RNA sequencing, where we look at a whole transcriptome assembly of both the coding and the non-coding RNAs. If we want to know how much of each transcript is present in a sample, you want to compare the transcript counts in one sample to another, we would do differential expression of mRNA sequencing libraries. And lastly, we may ask, and this is something that Peter also touched on last week with single-cell sequencing, you might want to know, how does transcript expression vary in heterogeneous samples? So in the same tissue might have different cells expressing different transcripts. And this is really important for something like cancer research, where you have tumors with differential subcolonies infiltrating the cells. Illumina has quite a few number of kits for this, so it's broadly divided into 3 applications. So the first one would be total RNA. This is where you maximize your discovery power, where you look at everything, your coding and your non-coding regions. So there we would suggest something like the new Illumina stranded total RNA prep, and this thing comes with a ligation. It's a ligation-based prep kit with Ribo 0 plus. The mRNA is a more cost-effective way, but this only looks at your coding transcriptome. So here we would use the poly A tail to capture the messenger RNA. One of your questions might be differential gene expression. You want to look at isoforms, allele-specific expression, fusion detections are possible and discovery, as well as detection of really low expressed mRNAs. And there we've got also a ligation-based kit, which is called the Illumina stranded mRNA prep. And then lastly, we might look at enrichment. So this is where you would design probes, and some of these kits are, for example, the viral pathogen kit, and then we've also got exome, RNA exomes that we can look at. So yeah, we've got a specific RNA prep with enrichment kit that we can use for that. So how do we choose a library prep kit for your application? So you're going to ask, what's my RNA-seq experimental requirements? what is your application? Do you want to look at coding RNAs? Do you want to look at total, meaning coding and non-coding? Or do you maybe want to look at your small RNAs? What is your sample input? Do you have really high quality inputs, or do you maybe work with FFPE samples, which will have a bit of degraded RNA in there? And then what is your budget? So does your budget allow for whole transcriptome sequencing, or should we then rather look at more targeted approaches? Your throughput, so how many samples do you have? And depending on the number of samples you have, we would have to choose the appropriate sequencer. Your data quality, what sequencing depth do you require? And would you require any replicates? And directionality is another question. You want to know, do you want the stranded or the non-stranded information for your RNA? So with the introduction of the Illumina RNA library prep suites, we've got a really fast workflow compared to the older kits. So it's a single day prep for whole transcriptome and coding transcriptome studies. It's a really high performance. It's well compared to the TruSeq, the old TruSeq kits. These TruSeq RNA kits have been cited in nearly 10,000 publications. Your input for these new kits are really low. It's as low as 1 nanogram for whole transcriptome sequencing and 10 nanogram for your coding transcriptomes. All these kits are automation friendly. So if your throughput is quite high, we can think about automating the process. It's got flexible applications. So we've got a single tube for rRNA depletion. And some of these kits are also FFPE compatible. And with the 384 unique dual indexes, we eliminate crosstalk between samples and we obviously support in higher output because of the high number of indexes with a more cost-effective sequencing in there. So with the introduction of these kits, we have different kit sizes. So if you don't have a lot of samples, we have the 16 sample kit, and we've also got the 96 sample kit. And as I mentioned, we've got up to 384 unique dual indexes. So the stranded total RNA kit is compatible with FFPE as well as our enrichment kits. Your input for the stranded one is really low. It's anything from 1 to 1,000 nanograms. Except if it's FFPE, we would say you need at least 10 nanogram for FFPE samples. And if you just look at the hours and compare that, your hands-on time is anything from 2 to 3 hours, which is really little. So the workflow for both total and mRNA preps, we would either do an RNA depletion. So we would do that if we want to look at total RNA sequencing, or we would then capture the mRNA by a poly-A tail if we want to do mRNA sequencing. Then, as with all other library preps, we would go through a fragmentation step, and then we would go through cDNA synthesis with reverse transcription to convert our RNA into complementary DNA. And what is really nice about these kits is the fact that the cDNA reagents are included in these kits. So it's not something that you have to purchase separately. And then we would do a ligation of our indexes, and these libraries would undergo a PCR amplification step, and then we would sequence. So with your total RNA preps, you get a really clear view of both the coding and the non-coding transcriptome. You've got the flexibility for many study designs, and you maximize the discovery power. And again, it's a hypothesis-free broad dynamic range. If we look at this graph, we compared the different input amounts, so from 1 nanogram to 100 nanogram RNA input, and compared compared to the higher input, there's a really nice concordance in the amount of transcripts that were discovered. So if you don't have a lot of RNA to work with, this would be a really nice kit to use. The other thing that we looked at is our FFPE samples. So where we compared 10 nanogram and a total of 100 nanogram samples. And again, you've got that normalized coverage across both concentrations. And we also have, they compared the Illumina stranded total RNA prep kit, which is the new kit, to the TruSeq stranded kit, which is the older kit. And again, the newer kit uses much less. It uses only 10 nanogram of input, where the older kits required 100 nanogram of total RNA. And again, you can see a very nice concordance of the data between the two different kits. So you can see that the backbone of these two methods, so the TruSeq kit and your Illumina stranded kit, is basically the same, but with a significantly reduced number of hours hands-on time. So again, with the new kit, it's a single tube where you can deplete multiple species of ribosomal RNA. It's up to 40% faster with 50% less hands-on time, and it's a really cost-effective and scalable way of doing transcriptome sequencing. If we look at the mRNA preps, we've got exceptionally good poly-A capturing efficiency. We've got high coverage uniformity, and again, it's a hypothesis-free based method with broad dynamic range. If we compare the Illumina stranded mRNA kit to the older TruSeq stranded mRNA kit, you can use much less input. So we can use 25 nanogram input compared to the 100 nanogram from the TruSeq kit. And again, you've got brilliant concordance between the two different kits. So on the NovaSeq, just summarized here, when you want to do gene expression profiling, with the SP flow cell, which is your smallest flow cell, you can do up to 80 samples with 10 million reads per sample and 1,000 samples on the S4 with 10 million reads. If we want to look at coding RNA discovery, we obviously need a little bit of more reads, so we recommend 25 million reads for these type of studies. There you can then put 32 samples per the SP flow cell and up to 400 on the S4 flow cell. And lastly, if you're looking at coding and non-coding discovery, we recommend deeper sequencing with about 50 million reads per sample. Therefore, you can add 16 samples on the SP flow cell and 200 samples on the S4 flow cell. So if we look at the analysis workflow for RNA analysis, the first thing that we will do is we will start to demultiplex. So demultiplexing is where you will separate your reads based on their indexes so that each sample has the exact same indexes. And therefore, you separate the reads per sample into a different read file to generate your FASTQs. Then we'll go on to filtering. So we trim the reads by quality and sequence, and we also remove the abundant reads. So with ribosomal RNA, they're the most abundant RNA species. It's about 80% of total RNA is made up of rRNA. And even though you do a depletion step, you will still have some reads. So there are some applications that can remove these abundant reads for you. And then we will do alignment. So here we will align to a reference transcriptome. And we would typically want to use an application which is at a splice-aware aligner. And the output file of this would be your BAM or CRAMP files. We would like to quantify, so you would want to know if you're doing transcript expression, you would want to count the reads. We would want to normalize the counts to have a more accurate comparison. And then we would like to identify novel transcript and maybe some gene fusions. And finally, you can do reports, and these outputs depend on which application and pipeline you're using. So there's different reports for differential expression, gene fusions, discovery of novel transcript, and then there are several single cell pipelines as well. So this is just a broad summary of the kits that we've mentioned. So I'm not going to go into too much detail, but it basically gives you the RNA input amounts, the different kits that you can use, the reads per sample that we recommend, and then how many samples you can add, let's say on the NextSeq 550, the NextSeq 2000, and the NovaSeq, and then some of the analysis options available on Dragon, which is available on Base-Based Sequencing Hub. So the last application I would like to discuss is the TruSight Oncology 500, or simply TSO 500. So TruSight Oncology 500, it's a next generation sequencing assay that enables in-house comprehensive genomic profiling of tumor samples. It supports identification of all relevant DNA and RNA variants implicated in various tumor types. So this slide just highlights the completeness of the assay in terms of content, showcasing that it includes DNA and RNA variants, as well as tumor mutational burden and microsatellite instabilities. So we've got 3 different kits available for TSO 500. We've got the normal TSO 500 kit and the TSO 500 high throughput kit. These kits are compatible with your FFPE tissues, and whereas your TruSight Oncology 500 circulating tumor DNA kits, they're compatible with blood samples. So both of these kits have 500 or represents 523 different genes. The TruSight Oncology 500 and the high throughput kit also looks at 55 RNAs. Then you can look at small DNA variants, your copy number variations. With the RNA kit, you can do fusions, and with the ctDNA, you can do gene rearrangements. The first bunch of kits can also look at your splice variants, which obviously won't be possible with your ctDNA kits because you don't look at RNA. And then there's also your immuno-oncology biomarkers that you can look at for both of these kits. So with these, the TruSight Oncology 500 portfolio, you really enable comprehensive genomic profiling of a bunch of different cancer types. So we look at solid tumor, it's a pan-cancer design, and it covers all the relevant biomarkers in guidelines and that's been discovered in clinical trials. We can detect both DNA and RNA variants, except the RNA is not detectable in the ctDNA applications. And then we also look at that immuno-oncology biomarkers, including your tumor mutation burden and microsatellite instability markers. And there's also emerging biomarkers that's been included in these tests. So with the different kits, we've got different assays to support your FFPE tissues and the liquid biopsies. And together, we can provide unique tumor insight leveraging the same DNA panel design. So typically with the circulating tumor DNA assay, it's a non-invasive sample type. And this is a tool to interrogate your circulating tumor DNA in metastatic diseases. And it reflects option when tissue quality is not sufficient and gives you tumor genomic insight. So typically in patients where the tumor location is not really accessible, or due to the size of the tumor, or even a patient's health we would suggest rather to do the circulating tumor DNA assays rather than trying to get a biopsy of the actual tumor. So when we look at the TS0500 assay, we look at, there's different ways to look at biomarker coverage. So there's your single gene assays where you would really require multiple workflows to look at the single genes. And this obviously reflects algorithms extend your turnaround time, and you can miss really important alterations with these single gene assays. We've got some hotspot panels, such as the focus panel. So these focus on your more prevalent alterations, but again, you could potentially miss really important biomarkers. So therefore, they've designed the TS0500 panel, which provides the full coverage of your coding sequences, and it identifies most of the relevant alterations. And this is just an example of what I'm talking about. So something like the hotspot coverage, you only cover the selected regions of the genome, and therefore you can miss actionable small variants of unimportant fusions. Whereas the TS0500 assay, you're looking at the whole coding sequence that is covered, and you can identify all relevant alterations, including single nucleotide variants, indels, copy number variations, fusions, and splice variants. If we look at the workflow for these three different kits, as mentioned, with the TS0500 and the TS0500 high throughput kit, we start off with FFPE samples, and with the circulating tumor DNA, we start off with blood samples. We do extraction of the nucleic acids, and then we would put them through the library prep and enrichment. So these would obviously depend on what your sample type is. And then we would choose the right sequencer for our application. So for TS0500, it's compatible with the NextSeq 550 and the NextSeq 550 DX system, whereas the high throughput and the circulating tumor DNA assays are can only be done on the NovaSeq 6000 system. And then we would do data processing and variant calling with Dragon, so either the on-site or the on-premise server, and we can also access that on the cloud itself. And then there are some options for interpretation, which will give you a very nice report in the end. So we do that with Perion DX, Clinical Genomics Workspace. If we look at the different number of samples that can be sequenced on the different sequences, so on the NextSeq 550, we can use the TruSight Oncology 500 panel. We can do about 8 samples per flow cell, whereas with the NovaSeq, if we do the high throughput test, we can do anything from 16 samples on the SP flow cell up to 192 samples on the S4 flow cell. And with the circulating tumor DNA, we can do 8 samples on the S2 and 24 samples on the S4 flow cell. So with Perion DX, it's a really seamless workflow for turning your raw sequencing data into an easy to understand report for your typical clinician. So we've sequenced 523 different genes. Then it utilizes, the program utilizes, or the software utilizes literature and guideline information to determine your variant significance. And it classifies those variants in types by significance. And in the end, you end up with one customizable genomic report. If we look at the workflow, so the workflow entails you to prepare or first extract your samples. So this is specifically the workflow for the circulating tumor DNA. So we would then dilute and prepare these samples. Then we would do end repair and A-tailing, followed by the adapter ligation. Then we would need to do a ligation cleanup, and we would have to add the indexes. And then we do a first hybridization overnight. On the second day, we would capture those hybridized probes. We would do a second hybridization, capture again. Then we would amplify these regions. We clean up the library with the site selection. We quantify the libraries, which is optional, and we have to normalize the libraries and prepare them for sequencing. So all in all, it's a two-day workflow for the hands-on library prep. So just to summarize the things that we've discussed, it's high throughput sequences with a bunch of different applications that we can do on the NovaSeq. We've got the chromatin accessibility and methylation profiling for your cancer epigenetics, profiling of your tumor and microenvironments you can do by spatial transcriptomics, single cell analysis, RNA sequencing, protein expression, and T-cell receptor and B-cell receptor sequencing. For microbiome analysis, we can do both 16S rRNA sequencing as well as shotgun metagenomics. And for your tumor mutation profiling, we can do exome sequencing and whole genome sequencing. And with that, I thank you for listening. If you have any questions, please let me know.
Thanks a lot, Mariette. Ladies and gentlemen, if there's any questions, you can either unmute yourself to ask Mariette the question, or you can just put it in the chat if you're a little shy. While we're waiting for questions, I can also say that, and I see your hand is up, I'm going to get to you now. I can also say that your recordings will be made available in the coming few weeks. So then anything that might have been missed, you can catch that up. Anna, yes, you've got a question.
Yes, thank you so much for your very good presentation. What I want to find out is if I have samples, because I know you do sell these different platforms, right? But do you also offer sequencing services? Like, say, I can send specimens to you for sequencing for a service, for a fee.
So we don't provide the service, but we do deal with a lot of labs which we can always refer you to. Obviously, we know where all the Illumina sequences are situated. We know what labs do what type of analysis. So we can always refer you to the correct facility if that helps.
Okay, all right. Thanks.
Yeah, so you can just take down my email address and send me an email. We actually have a few NovaSeq sequences on its way to South Africa. As yet, up till now, we actually didn't have any NovaSeq sequences, but now we've got 3 of them coming to South Africa, so we're very excited about that. And that kind of keeps the sequencing in South Africa rather than people sending their samples overseas, which is really costly. And I think, all the logistics behind sending samples overseas, I think a lot of you can attest to it. It's quite tedious. So.
No, I actually didn't mean to send samples over, but tend to send to separations here in South Africa.
Yeah, so you can just let me know what type of samples it is, what exactly it is you want to do, and I can refer you to the correct lab.
Okay, thank you so much.
Pleasure. Any other questions? Okay, I don't see any other questions. Then I wish you a lovely weekend ahead, and thank you once again for attending this webinar series. We hope it was insightful, and yeah, we hope to see you soon again. And if you have any questions, please feel free to either WhatsApp us or send us an email. We're always here to help.
Thanks for your time, Mariette, and for the nice presentation.
Your pleasure.
Thank you, ladies and gentlemen. Bye-bye.
Thank you very much. Nice presentation. Bye.
Thank you. Bye.